Everyone, we'll go ahead and get started. Hello. I'm so glad you guys came again. This is awesome. You guys came and I'm really happy to see you all here. And thank you for coming, everyone. Yeah, this is our next in series of the stem cell community lecture series. And just to give a little bit of an overview about the Stem Cell Center, and I know everyone here has really heard quite a bit about it, but we just had Stem Cell Awareness Day last Saturday, and it was a really nice event and a lot of students that came through the center. The second Wednesday in October, we're having it coming up Wednesday, October 8th. That last Saturday, we had a stem cell community forum activity. So Stem Cell Awareness Day is coming up October 8th. And this was designated as Stem Cell Awareness Day by the California Institute for Regenerative Medicine and former Governor Arnold Schwarzenegger in 2008. And so what are stem cells? We're going to be talking about those today. Stem cells are fundamental building blocks, and they can keep dividing, and they can divide for very long periods of time, for basically forever. And we can turn them into any cell type that we want to study or use in therapeutics or understanding disease mechanisms. And it's really it's been a transformative technology that we've been able to utilize. I mean, I remember when I was starting, I work on Huntington's disease, and when I was starting in the field, we had no good cell models. It was just so difficult to study, say, try to use a cancer cell line, for instance, that would divide in a dish, and try to use that for studying Huntington's disease, which is a brain disease. But this really has enhanced our understanding of how diseases develop and how to treat them, more importantly, how we should go about treating them. And these can result in new treatments for all sorts of different diseases, blood diseases, brain diseases, diabetes, all sorts of things. And the center itself was dedicated in May 2010. And this was through a CIRM grant and also through philanthropy. And it was the first building in California that was designated as a stem cell center. So it's pretty exciting, and we love this building a lot. It's just been an incredible place for collaborative research, for interaction among colleagues and learning from each other. And we're very, very grateful to California voters and people who voted this in. And right now there are 116 CIRM-funded clinical trials, as I mentioned, all sorts of applications throughout California that are CIRM-funded. Our Alpha Clinic here that does cell and gene therapies has 25 active clinical trials. And we didn't have any, not too many years ago. And now we have 25 active clinical trials with over 200 patients enrolled to date, and that's continuing to grow. And there we have a very active training program for students here at the Stem Cell Center. We have 9 NIH and CIRM funded training programs here in the Stem Cell Center, which is amazing. And so we can train anywhere from undergraduates up through graduate students and postdoctoral fellows in their as they go along in their careers and learn about stem cell applications in biology. And just a quick overview, there's 65 faculty in the center. It represents 27 different academic units throughout University of California, Irvine, 7 different schools. So say the School of Medicine has multiple departments. That's one of the schools involved. And we have, as I mentioned, the Alpha Clinic. We have quite a bit of funding from CIRM over the year, $178 million far. And we have gene therapies that are moving forward for these various disorders: diabetes, blood disorders, spinal cord injury. Brian's taking one for traumatic brain injury. So I should actually have introduced myself. Leslie Thompson. I'm the new co-director with Brian Cummings for the Stem Cell Center. We've also funded the center's funding seed grants for individuals in the Stem Cell Center, but there's also been a lot of different seed funding grants that have emerged from the CIRM funding over the years. $20 million in research funding resulting from those seed grants that were just started as very small, $50,000 type grants, $25,000 grants, that then we're leveraged to get all sorts of other types of funding. So it's really been an incredible center. There's right now 65 total research grants, around $24 million of research going on in the building. So we're very excited about this. And these are some websites that if you're interested. And it had our Stem Cell Center website has upcoming events, has latest reporting on various activities that are going on. So if you're interested, feel free to go to the site. So without further ado, though, I'd like to introduce our speaker for tonight, which is really a pleasure. Yongsheng he has been at UCI since 2008, right?
Yes.
Yes, started here as an assistant professor in microbiology and molecular genetics in the department within the School of Medicine. And he also runs now, launched and directs the UCI Center for RNA Science and Therapeutics here at UCI, which is really outstanding. There's a lot of individuals here on campus that work on RNA biology, but he also brings in a lot of outside colleagues and speakers here. And I think you can pretty safely say, maybe except from the very, very beginning, that you have been such a leader in RNA biology and the dynamics of how RNAs work in our cells, how they transmit information, how they get modified by, and I'm sure he'll talk a little bit about polyvenylation, but I'll let him talk about that. But he's really a leader in this whole area of RNA biology, which we've realized over the years, it has such a critical role in regulation of everything that goes on in the cell. So without further ado, Dr. Shi, thank you.
All right. Thanks, Leslie, for the introduction, and thank you all for coming. So my name is Yongsheng Shi. As Leslie mentioned, I've been a professor here for quite a long time, and I originally came from China, and it's been a blessing to be able to go to graduate school and postdoc, and then do research here in the States. I think in these days it's becoming more difficult. It's harder to get into college or graduate programs in the US. And then after that, it's hard to, even if I get a job, nobody is going to pay $100,000 for an H1 visa for me. So it's going to be, hopefully things will turn around. But fortunately, we have you guys. Hopefully you will pick up the torch and then really do become scientists in the future. So I'm told there's a lot of high school students. So which high school are you guys from? Okay, all same high school? All right, great. And which high school are you guys from? Right. Joking.
Last year.
Yeah,
From Eastminster.
yes. Yeah, so today we're gonna talk about some stem cell work and the role of RNA in stem cells. And so there's gonna be a lot of so let's start from the basics.
Are you gonna have any
there's
dark slides?
No, it's fine. It's fine. So let's start from the basics. And so as Leslie mentioned, so we all have stem cells. All of us started from one cell, the fertilized egg, right? And from that one cell developed into our whole body. So that one cell can basically differentiate into, so at the beginning, fertilized egg is called totipotent. What that means is that one cell can become any cell type in our body, right? And then as they differentiate and become more and more cells, these cells become more and more specialized. So for example, as you get to the embryo stage, then now here some of these cells are embryonic stem cells. So these cells are called pluripotent. So they can become almost all of the cells in our body except for placenta. So that's basically the tissue outside of the embryo, right? So yes, embryonic stem cells cannot become placenta. Other than that, they can become any cell in our body, all right? And then embryonic stem cells during development, then it becomes more and more specialized. So now you have, for example, hematopoietic stem cells, which later will become developing all kinds of blood cells. And then you have epidermal stem cell, mesenchymal stem cells, neural stem cells. For example, from neural stem cells, you can develop all kinds of neural cells in our brain and our peripheral neural system. And so basically, from this one totipotent stem cell, we get all these differentiated cells. So then one interesting question is, as the cells become more and more differentiated, it seems like their capability becomes more and more limited, right? As I mentioned at the beginning, it can become any cell in our body. But then later, if you, for example, if you take a skin cell, even if it proliferates, it's going to produce more skin cells. It's not going to make, for example, neurons or other type of cells, right? So seems like the cell during differentiation, they lose some of the ability to become certain lineages. So how does that happen, right? So that's a very fundamental question. As in history, people basically started wondering about these questions very early on. So in the 1880s, so this guy, August Weismann, proposed this very famous hypothesis. So his hypothesis was, during stem cell differentiation, the cells will gradually lose some of the DNA in our genome. So because you lose these genome, the DNA, then you lose the information stored in these DNA pieces, therefore you lose the ability to become certain type of cells, right? So that sort of makes sense. And so that's the original proposal, but then somebody actually did an experiment to test that idea. So the experiment was like this. So here they took a frog egg, a fertilized frog egg. So then from this one egg becomes 2 cells, 4 cells, 8 cells, and becomes the full frog, right? Or tadpole at the beginning. So what this guy did was they took this embryo at the 2-cell stage. So basically, the 1 cell just divided into 2 cells. And then they took one of the cutting edge technology at the time. Basically, he used the hot needle to poke one of the cells, right? So basically, because a hot needle, you poke the cell and you kill that cell, right? So now you have 2 cells and you kill one of the cells and then wait to see what happens to this embryo. And interestingly, when they wait until the end, what they found was that this embryo becomes half of a frog. So basically, you get one half of the frog, the other half is kind of just a mess. And so from this, he took that as evidence that this proves that his idea, which is called the germ plasm theory, or another name is called the mosaic theory. So basically, in our body, all the cells are basically mosaic. So they're not the same. Every cell has different DNA. Every cell has lost part of our genome. Therefore, they cannot develop into some type of cells. So this is the original theory in this area. And then a few years later, somebody else proposed a completely different theory. So this theory is called regulatory development theory. So in contrast to the first theory, where they proposed that during development, each cell just lose DNA gradually. So in this theory, this German biologist Hans Driesch proposed that actually all of our cells keep the same DNA from the fertilized egg. But all that happens during development is that during development, different genes are turned on. So different cells will turn on different sets of genes. So therefore, you become different kind of cells, right? So that is his theory. So the way that he actually did the experiment to test it, so here instead of frog, he used a sea urchin. Again, he got this 2-cell sea urchin embryo. And then instead of using hot needle to destroy one of them, he actually physically separated the 2 cells. And then keep growing each cell to see what happens. So interestingly, each cell becomes a full sea urchin, right? Identical. Actually, no DNA was lost. So each cell has the full capacity to develop into a full animal, suggesting that no DNA is lost, right? So now you see this you can see that these experiment was done in the 1880s, and these experiment was done in the 1890s, right? So you have these conflicting results coming out around the same time. So what do we believe?
Let's have a wrestling match.
Yes, yes. So this, of course, the people can say, all right, these are sea urchins, right? So they are more primitive, whereas the frogs are vertebrates. So maybe the primitive animals, they can keep the capability to each cell can develop, but maybe in higher animals or cells, so you lose the ability, right? Maybe they use different mechanism. So then how do you test this? So actually, there's other than frogs and sea urchin, from other species, we see examples of cell fate changes. Basically, cell lineages can change from one to another. So one very familiar example to all of us is the earthworm, right? So if we get an earthworm, you break it into two halves. Each half will fully develop into a full worm again, right? So you've seen this, so you can. So this suggests this is kind of consistent with the sea urchin model, where each cell keep the full set of DNA. They don't lose any DNA. Because here, even if you just have the tail, you can still grow the head. So that means the genes required for head development is still present in these part of the body, right? So suggesting that the DNA is not changing. So this is earthworm. Again, it's very primitive. Let's look at a more sophisticated organism. So this is salamander. All right, salamander is well known to have this capability of limb regeneration. So what you can do is you can actually cut off the limb of a salamander, and over time, the limb will grow back. And so this is, I don't know if you guys have seen the similar vertebrates. Sometimes you can they can break off their tail and the tail will grow back, right? So all of this suggests that even vertebrates actually has the ability to change cell fate. So basically one cell type can change to another cell type. For example, here it lost the digits, but then the tissues in arms, then they somehow took on the new identity of these digits. So therefore, suggesting that the cells don't really lose any DNA or don't lose the capability to become other cell types. It's just that you turn on different set of genes in different cells. And in fact, this is salamander, but even in human, I've read reports saying that we can also regenerate limbs, maybe not the full limbs, but some kids, little kids, if sometimes their fingertips are broken off by accident, and they can actually grow back the full fingertip. So, but of course, once you become adult, you lose that ability, right? So suggesting that there's some flexibility in this kind of cell fate change. So of course, we can see that there's different evidence from worm, from frog to reptiles. So they seem to suggest that all of our cells keep the full set of DNA and it's fully capable of developing into any tissue in our body. But that's kind of indirect evidence, right? How do you directly test it? How do you know for sure that each cell has the DNA that can fully develop into any cell type, right? So what is the experiment to test this? So this experiment was done by John Gurdon, a British biologist, in 1962 using frogs. So what he did here was he took the small intestine of a frog and then break up the small intestine into individual cells, and then use this fine needle to take out one of the nucleus from these cells, right? So the nucleus is where the DNA is stored, right? So you basically take the nucleus that contains the DNA from this one single cell, and then he took an egg cell from the frog, and then remove the nucleus from the egg, right? So now this egg does not have DNA anymore. So then what he does is he inject this nucleus from this intestine cell into this egg cell. So now this egg receives a new nucleus. So this nucleus was, remember, originally from the intestine, right? So now, once you put this nucleus in this egg cell and then let it develop, it turns out this can develop into a full frog. So what that means is that the DNA contained in that the intestine cell contains all the information you need to develop into a full animal. All right, so this is the first definitive evidence to show that all of our DNA, even in the fully differentiated cells, contains all the information, all the genes that's required for development. And so this is actually the picture from so he obviously didn't do just one, he picked many nuclei, injected many eggs, and got many frogs. So this is the frogs cloned from this frog. And so this is John Gurdon. Actually, sadly, he passed away today.
Today.
Yes. Yeah, just saw it on Twitter. So he passed away today, and he was one of the pioneers in developmental biology. So he showed the first definitive evidence that all of our cells contain the DNA that contain all of our genes. And so this is an amazing experiment. So then, of course, this was first done in frog. So once people realized this can be done in frog, how about in other species, right? So then people started this frenzy, just tried to clone all kinds of animals. And one of them is Dolly the sheep, right? So in 1997, you all remember this in the news. So this scientist in Scotland, so basically, again, that's doing very similar things as John Gurdon did in frog. So he took a differentiated cell from a sheep and took out the nucleus, and then he took out an egg from another sheep, removed the nucleus, and then put this nucleus into this receiving egg, and then used this cell to develop into a whole new sheep. So that's Dolly, right? So this, obviously, the Dolly has exactly the same DNA as the starting sheep. So actually, 2 years ago, I visited Scotland, and so actually this is in their science museum. So when Dolly died, so they made it into a specimen so we can all look at.
Oh, wow.
Yeah, so this is again, this is an important piece of history. So now not only you can clone frogs, you can clone mammals, right? So then people have by now pretty much cloned everything you can imagine. So there's panda, dogs. So people even try to develop into a business, right? So if you have a dog that you love so much who's passing away, you might be able to clone the dog and make a whole new dog that has exactly the same DNA as your old dog, and then the dog can live on. Or there's other applications where you have endangered species. For example, a giant panda, right? So it's going extinct. So how do we preserve them? So maybe we can just try to clone them, right? And there are even cases where people try to revive extinct species. So that's the whole Jurassic Park, right? So you can take the DNA from the extinct species and inject it into the dinosaurs or reptiles. So maybe you can take that DNA from the reptile and inject it into from the dinosaur and inject it into a reptile and try to clone a new dinosaur from it, right? So either way, so all of this is suggesting that all of our DNA remain intact during development. Of course, now we know that there are exceptions in some blood cells, for example, B cells. They do lose some DNA when they make, for example, antibodies. But in the vast majority of our body, all the cells contain exactly the same DNA. The only difference between tissues is that different tissues express different set of genes, and that's why they look different, they function different, and so then from here, we show that this can be done. So you can reverse development, right? So you can take a fully developed cell and induce it back to the stem cell totem state. But here it's quite complicated. You take the whole nucleus, put it in an egg, and let it develop. So what if we can just take any cell and don't transfer the nucleus, just directly induce it to become a stem cell like a cell, right? So if that's the case, it becomes much easier and has much broader applications. And so this was done actually in 2006 by this scientist called Shinya Yamanaka. So he was trying to do this impossible experiment. So after people have done this nuclear transfer, we know the development can be reversed, right? And so now he's trying to do this crazy thing. You can take a cell, differentiate a cell, you can take a hair follicle cell, any cell, and then just directly make it into a stem cell. How do they do that? Well, like I said, the idea is stem cells and differentiated cells, they're simply expressing different set of genes, right? So stem cells must express a set of genes that make them stem cells, whereas differentiated cells don't express these genes, right? So in order to change a differentiated cell back to a stem cell, all you need to do, theoretically, is to induce the expression of the stem cell specific genes, right? So now, if a differentiated cell is beginning to express stem cell genes, it will behave like stem cells, right? So that's the idea. But the challenge is, if you just compare stem cell versus any differentiated cells, the gene expression changes the gene expression patterns are vastly different. There are at least hundreds, oftentimes thousands of genes that show different expression patterns. So how do you know which stem cell specific gene is able to convert differentiated cells back to a stem cell, right? So that's like looking for a needle in a haystack. But he actually did the impossible. So what he did was he compared stem cells versus a whole bunch of differentiated cells, and then found a group of candidate genes that are only expressed in stem cells and are not expressed in not any of the differentiated cells, right? So you come up with a list of genes. At the beginning, it was about 50, and then later it was narrowed down to 20. So then he just tried to express one gene at a time. So you take a skin cell, you express this one stem cell specific gene in the skin cell and see if it can turn it into a stem cell. And if you have 50 candidates, you just express 50 genes, one at a time. But none of that worked. If you just express one gene, it doesn't convert the cells back to stem cells. So out of desperation, what he did was basically dump all 50 genes into the cell and see what happens. And then we came back and saw they're actually stem cells growing, right? So typically when you have these skin cells, after a while, they stop dividing. And then he dumped all 50 genes in there. Weeks later, there's still cells growing. So those become stem cells, because if they were not stem cells, they would have stopped growing, right? So then he narrowed down from 50 to 20, and eventually he narrowed down to 4. And finally, the killer experiment was that it took a fibroblast, or actually later people show you can take pretty much any cell in your body, and then just overexpress 4 genes. These 4 genes, OCT4, SOX2, KLF4, and c-Myc. If you express these 4 genes, you will get stem cells. It's basic from any cell type. So when that study came out in 2006, that really shocked the world because nobody thought that was possible. Just 4 genes is enough to bring any cell back to a stem cell-like state. Because originally we had to transfer the entire nucleus, but now it's just 4 genes. And so this is the stem cell made this way. It's called induced pluripotent stem cells, or iPS cells. So the reason why it's called induced is because it's induced by the expression of 4 genes, right? And induced pluripotent stem cells. So they behave like embryonic stem cells. Basically, they can become any cell type in our body. This really opened up a whole new area of research and make possible a whole bunch of applications. So you can imagine that how we can use iPS cells. So now oftentimes on campus, we have blood draw, right? So basically, there's a truck there's a bus on campus that people should go there to donate blood. So we collect blood from donors and then keep them in hospitals. And then when somebody needs blood transfusion, you match the blood type, you then provide the blood. But using this technology, you can imagine, what if, for example, we can take some cell from our body, right? And then make it into iPS cells, which again, like stem cells, it can become any cell type we want. So then we can differentiate those cells into blood cells, right? And then we can put those cells back into our body. And then we don't need to rely on donors anymore. And that you can also, the advantage is clear, because for donor, often even for just simple blood transfusion, you have to match the donor type, blood type and your blood type, because otherwise you're going to have immune response, right? But if you take your own cells and convert it into blood cells, there's no immune response because that's your own cell, right? And so that's just one potential application. So here, basically iPS cell, you can differentiate into any cell you want. And then you can make into neurons. So for a lot of neurodegeneration diseases, so for example, Parkinson's, Huntington's, all these diseases, and basically you have neurons that are dead, right? So they're damaged and they die, and then you have all these neurodegeneration disease. So what if we can take the cells in our body, convert it into iPS cells, and then differentiate that cell into neurons, then put that back in our body, right? So that can potentially treat the disease. And so this is the direct application in therapy, but it's also, as Leslie mentioned at the beginning, it's also extremely useful for research. Because if somebody has, for example, if we want to study Alzheimer's disease, so how do we study that? The patients, each patient is different mutations can cause Alzheimer. Every patient, the driver may be different, the phenotype might be different. So basically you can have different causes and you have different severity. It's very difficult to study this. So how do we, if you make an animal model, so mouse, they don't look like human, they don't behave like human, the mouse neurons don't, oftentimes they're different than humans. So with this technology, what we can do is that you can take the brain cells from the patients, right? And then you can convert them into iPS cells. And then you can differentiate those iPS cells into any neural cell you want that you think might be relevant to Alzheimer's disease. And then you can see what happens, right? So you can have a patient cell, you can have a cell from a healthy donor, right? No mutations. So then you can do the same thing, induce both into iPS cells, and then differentiate them into neurons, and then compare the two, right? So the neurons derived from healthy donors versus the neurons derived from Alzheimer's disease patients. See what are the differences, right? Those neurons have the same DNA as the patient. So whatever you find is going to be directly relevant to the patient. So those are hugely important tools and models for studying disease. And of course, once you have a good model, you can also use that to screen drugs. So for example, you have again, using the Alzheimer's disease model, you make a neuron from the brain cells of the patients. Now, for example, if it grows poorly on a petri dish, right? So that's a model. Then you can use that to screen for drugs. You can add different drugs to the cell to see which drug can make the cells grow better, right? And then of course you can't do these on patients directly, but by using these cell system, the iPS cells, you can do this, and the outcome is directly relevant to the patient. All right, so here, obviously, this opened up an entire area, and it's actually as soon as this technology was developed in 2006, many people realized this. There are even people that immediately start businesses freezing down blood cells from umbilical cords from babies, right? So then the idea is those are stem cells from babies, and then you can maybe use those cells to differentiate into any cell you might need in the future, and you can use that to treat diseases. And so here, this technology is very powerful and it's very promising, both for basic research and for clinical applications. However, there are still a lot of bottlenecks. One of the bottleneck is that this process is very inefficient. So what that means is, so here, so typically you start with a whole bunch of cells, right? And then you overexpress these 4 factors. So those are the Yamanaka factors, the 4 genes. And then you just overexpress them and you wait and see how many cells become iPS cells or stem cell-like cells. So typically, in different species, this process takes different amount of time. If you take a mouse cell, overexpress this 4 genes, it takes 2 weeks to get iPS cells. But if you take human cells, it takes much longer. And not only it takes a long time, but also the efficiency is very low. So for example, in mouse, if you take a large number of cells and then you express these 4 factors, wait for 2 weeks, in the end, about 0.1 to 0.2% of the cells becomes iPS cells. So that means if you take 1,000 cell and do this, maybe 1 will become iPS cells. The rest will be stuck somewhere along the way. They might be partially reprogrammed, but they're not fully induced pluripotent stem cells. So why is that? Well, as we mentioned, different cell types express very different set of genes, right? And if you want to drive the gene expression pattern to stem cell-like pattern, so that it doesn't, you don't achieve that in one step. So basically, people hypothesize and a later experiment showed that you achieve this in several steps. So basically, during the expression of these 4 factors, one set of genes will change first, and then later another set of genes will change later, and then gradually you become more and more like stem cells. However, not all cells will be able to finish all these transitions. Some cells may change the first wave of genes, but then cannot change the second wave, right? So they're stuck. So basically, they're partially reprogrammed, but not fully reprogrammed. And that suggests that this whole process is actually very difficult. And why is that? Well, the one idea is that maybe our cells don't want it to reprogram easily, right? Which actually makes sense, because if you have a skin, for example, if you take your skin cells, once the skin cells become skin cells, you want it to remain as skin cells because it has specific functions there, right? You don't want it to easily change to another cell type. And so one of the hypotheses that maybe there are mechanisms in our body that prevent reprogramming. So after the cells have become a specific cell type, there are mechanisms to lock in that state and prevent them from changing to other states. And so those mechanisms, so we believe there are some genes that can do that prevent reprogramming. And so those are called roadblock genes. So if you look at this change, if you think of it as a step-by-step pattern, each step may represent a group of genes that's preventing this step, right? So only some cells will be able to step up and overcome this roadblock. And then another roadblock until it gets to the final stage. So what are these roadblock genes? And how do we find them? So that's the question that my lab became interested in. And so here to look for these roadblock genes, so what we did was, well, the human genome has 20,000 genes. Any of these genes could be a roadblock gene. So how do we know which one is? Well, if we don't know which one, and we don't want to guess because there's 20,000 genes. So what if we can test all of the genes in an unbiased way and figure out which gene may be involved in this, right? So this is to do that, we use this technique called genome-wide screen, genome-wide RNAi screen. So what it is there are tools that allows us to decrease the expression of a single gene, so one gene at a time, right? So we have 20,000 genes. All we need to do is make 20,000 different siRNAs. So these are RNA tools that allows us to target one gene at a time. So if I make a siRNA for gene A, I introduce that siRNA into cells, it will bring down the expression of gene A. Then we can make another siRNA for gene B, and will specifically bring down the expression of gene B, right? So then what we do is we make 20,000 of these siRNAs. Each one targets one gene in our genome. So then what we can do is we can take the differentiated cells, let's say skin cells, right? You can get tens of thousands or millions of cells. And then you introduce these siRNAs into the cells. So ideally, you have one siRNA in each cell. So then within that cell, only one gene expression is reduced, right? Once we reduce that gene expression, then we try to reprogram this cell. So typically, like I mentioned, 1 in 1,000 cell can be reprogrammed. So the likelihood is very low. But what if there are some genes, when you knock it down, the cell becomes easier to reprogram, right? So especially if it's a roadblock gene that prevent reprogramming, if you remove that gene, so now the cells becomes easier to reprogram. So then what you can do is, so we took these millions of cells and then introduced these siRNAs into these cells. And then we reprogram them. After that, we collect the iPS cells. And then from there, we just look at these siRNAs and see which siRNA they express. And then we can figure out which gene in these cells were actually suppressed, right? So that allows these cells to become reprogrammed more easily. So by doing that, we figured out that the top candidate was this factor called CF125. So I'll show you the results. So here is the results when you do a reprogramming assay using mouse cells. So if you take a mouse cell and grow, let's say it's skin cells, you grow it on plates, it will form this very flat layer of cells. But then once you overexpress those 4 Yamanaka factors, and then wait for 2 weeks, so some of these cells will become iPS cells. So most cells, after 2 weeks, they stop dividing, they don't grow. But if one of these cells become an iPS cell, so now they can grow. Theoretically, they can grow indefinitely, right? So they can grow and form these little dots. These little dots, each dot is a clump of cells. Because stem cells can grow, so 1 becomes 2, 2 becomes 4, and then they grow to form these big clumps. So each clump here is called a colony of stem cells. And so there's also a specific dye that you can use to dye these cells, and if they turn red, they are stem cells, right? So now, but if you stain these cells, you can figure out how many iPS colonies there are, how many cells are reprogrammed. So here is just straight up fibroblast cells reprogrammed for 2 weeks. You get a few colonies, not too many, but you do get them. And then if you use this control siRNA, which doesn't knock down any gene, and you also get some colonies, but not too many, similar numbers. However, if you knock down this gene that we found from the screen and then do the reprogramming, so now you can see you get large increase in the number of IPS colonies, right? So that means, so this is actually 30-fold increase in the number of colonies. So what that means is, so typically it's the efficiency is very low, but if you knock down this factor in the cell first, differentiated cells first, then reprogram, you get about 30-fold increase in the reprogramming efficiency. So that suggests that this factor is a key roadblock gene that prevent reprogramming of differentiated cells. So if you inhibit it, it becomes easier to reprogram, right? So then we later figured out the actually how it works, and so I'm not going to get into that. But in terms of application, this finding is potentially useful because, like I mentioned, if you want to use iPS cells in therapy, right? So you want to take cells from patients, convert it into iPS cells, and then use that to treat diseases. So you want to be able to efficiently make iPS cells. If 1 in 1,000 cells, and sometimes even less than that, becomes iPS cells, it's very difficult to use that technology, right? So then, what if we can inhibit this factor in cells before we do reprogramming? Then we can increase the efficiency dramatically. And of course, here we're using an siRNA, put that into a cell to reduce the expression of this gene. But that's not very, potentially not very safe and difficult to do, because usually you have to, this siRNA, usually it's packaged into a virus-like particle and put it into the cells. And so it's difficult to do, and the effect may be more long-term. So it's not, we only want to inhibit this expression of this gene for a few days, right, during the reprogramming. We don't want to inhibit it for the long term. But if you use siRNA, it's very difficult to control how long you inhibit it. So how can we inhibit it very quickly, very efficiently, but we can stop it anytime we want? Well, the way to do it is to develop a drug that can target this factor. So this factor, so here is the crystal structure. So this is the protein. It forms a dimer, and it binds to RNA and regulates gene expression. So here is, you can see there's this little channel, a very narrow channel, and this is where the RNA comes in. And this protein binds to RNA through this channel, and then have its functions. So to inhibit this, remember that in previous slide, we were knocking it down with siRNA, but that's not very good. So what if we can develop a small molecule drug, right, a chemical, that we can add into cells? Those cells will bind to this channel. Once those drug can bind to this channel, this protein cannot bind to RNA anymore, right? Because the channel is occupied by the drug, so therefore it cannot bind to the RNA. So that drug essentially will inhibit this protein. It's equivalent to if you knock it down, like if you inhibit its activity. So if we can develop a small molecule drug like this, then you can imagine what we can do in the future is you can take patient cells and add this drug, right? And then you induce reprogramming. So after a few days, so that the boost in the efficiency is already accomplished, then you can remove that drug. So now it's already well on its way to be fully reprogrammed. We don't need to inhibit that factor anymore. We can remove it, right? So then you can basically get the benefit of higher efficiency in reprogramming without the long-term potential harm. So we can accomplish with a drug like that. So now we're developing assays to screen a large number of chemical compounds to try to find ones that can inhibit this protein very specifically. Once we can do that, we can potentially use that as a tool for iPS cell technology. And so this is basically what I'm going to talk about today. And so my lab consists of a bunch of very energetic students and postdocs and technicians. And I do host high school students in the summer. And so it's a lot of fun. We work on not only stem cell related projects, but also a variety of other projects that are related to RNA. And so now, before the pandemic, whenever I tell people I work on RNA, it's always very difficult to make it clear what I do. So my parents never understood what I do. But after the pandemic, at least they know RNA. So even if they don't know what they look like, they know what it can do. So now I think that there are a lot of potential in using RNA and RNA targeting proteins as research tools and in treating a lot of diseases. And for example, this is one example, right? How you can target a RNA binding protein and to make a better system for making iPS cells. So that's my research. So I forgot to take short breaks in the middle, but I think hopefully I'm talking slow enough that now I'm open to questions. Thank you for your attention. All right.
If you're able to inject an IPS, can you extract those bad cells as opposed to constantly keeping re-injecting? And my second question is, if I look at drugs are after the fact. Okay, is anything being done on prevention before it actually occurs? I mean, any kind of screening method to prevent, let's say, Parkinson's or Alzheimer's or whatever, as a result of this research.
Yeah, so there's obviously iPS cells can be used potentially to treat a lot of diseases, but in terms of extracting the bad cells, that is a difficult challenge. So I think the first application area is the area where, for example, if you have cell death, right, in neurodegeneration, some cells are dead and they're not functional, and we can replace those with functional new cells. But those cells may not need to be cleared away because they're already dead and where you have, for example, in cancer, you have constantly bad cells growing, and you can't just replace those with good cells. You have to prevent those bad cells from growing. That requires a new set of tools in addition to iPS. So I think for iPS, my understanding is probably right now the immediate application is, for example, in brain and these diseases, but maybe not so much for cancer, where you need to get rid of a lot of bad cells.
Thank you.
Yeah.
Why is it that CFLM25 like inhibits the iPS growth?
Yeah, that's a great question. So I have prepared some slides to answer your question.
Just in.
Yes.
He's not a plant.
Yes. So in high school, you learn about gene expression. So you have genes, right? So they are transcribed into RNAs, and the RNAs are made into proteins. And so here is a gene that's made into RNAs. So these boxes are the blue boxes are the protein coding sequence, right? So here from left to right, the sequence starts from here and here that encode a protein. However, in most of these mRNAs, after the protein coding sequence ends, there are extra sequences before the poly A tail. So all the mRNAs have a poly A tail, have a protein coding sequence, but there are also these sequences in the middle. And so those don't code for any protein, but they have functions in regulating the expression of the mRNA. And interestingly, in our genome, for most of the genes, you can polymorphize the RNA at different places, so that you can, for example, in this case, from one gene, you can make 3 different types of mRNA. And all these mRNAs encode the same protein, but they have these different sequences. So these are called the 3' untranslated region. So although they're not translated into proteins, but they regulate gene expression, the expression of these mRNAs. And by and large, the longer these untranslated regions are, the lower the expression from this mRNA. And that factor that we found is actually regulating this poly-A site choice. So basically, what we found was, so this is a factor that we found in the differentiated cells, right? If you knock it down, you get a higher efficiency of reprogramming. So then from those cells, obviously we want to figure out how it does it. The way we did it was we took the cells, either regular cell, differentiated cells, or cells, we knock down this factor, right? And see what happens at the RNA level. And we actually found many changes. And by and large, if you can see, most of the changes are red, right? So they're basically 10 times more red dots than blue dots. And it turns out what this factor does is, in the differentiated cells, typically most of the RNAs have a very long 3' untranslated region. And remember what I told you earlier, the longer this region is, the lower the expression is from this mRNA, right? But if you knock down this one factor, this untranslated region becomes much shorter for hundreds of genes. And so just because of this, the expression level for these genes go up. And so these genes happen to encode some of the chromatin modifiers and other regulators, so these factors, you have to decrease their expression, you have to increase their expression to reprogram the cells. And by knocking down this factor, by regulating the untranslated region, you increase the expression of these genes. These genes, the higher expression of these genes promote reprogramming. So basically this one factor regulates hundreds of genes through this mechanism, and through this hundreds of these genes, you get higher reprogramming efficiency. So that basically that's where RNA comes in, and it's actually not. Remember we talked about the differentiated cells or stem cells. They express different set of genes, and in this case, a lot of the genes are expressed both in the stem cells and differentiated cells, but they're expressed at different levels. And it's regulated at this level. So basically how long the tails are. And in differentiated cells, the tails are longer, and in stem cells, the tails are shorter. And that is regulated by this factor. So therefore, by manipulating this one factor, you can change the reprogramming efficiency. All right.
Thank you.
When you think the nerve cells get converted into like the stem cells, can you kind of clarify the process of how they get differentiated into different neurons for like different areas of the brain, like the amygdala or the hippocampus?
Yes, so that's actually the expertise of Leslie's lab. And so basically, you can take iPS cells, right? So the iPS cell, you can take any cell, express these 4 factor isolate iPS cells. So now there's even for humans that we have, sometimes you can take patient cells, and there are also established iPS cells available for general research. You take these cells, and there are ways to differentiate them into different types of neurons, for example. So the way to do it is you provide different growth factors. So that will induce them to go down a certain pathway, right? So if you provide this growth factor, they may become neurons. If you provide a different growth factor, they may become astrocytes. And so it all depends on the growth factor that you put in the media. So you grow these cells in these media using these different growth factors, and that will drive them to different type of cells. Okay? Yeah.
Where do you see yourself in 10 years with this idea?
Yeah, so this is, I think Leslie may be the better person to answer the question.
What was the question?
So where do you see the iPS cell technology in 10 years?
Oh, that's a very good question. I think that there's gonna be a lot of applications for disease therapies from it. So for instance, exactly like Dr. She was talking about is where you can replace brain cells for something like Parkinson's disease, exactly, for instance. And some of the trials that are going on right now are using what are called embryonic stem cells instead of from patient samples, but embryonic stem cells that also can differentiate along any lineage. And they're taking like a single cell line and making those into precursor for the neurons that die in Parkinson's disease, for instance. And so, but it's a single cell line, and so people have to be immunosuppressed because they'll react to that cell line. But it's starting to show some promise for Parkinson's disease, for instance. But the ideal scenario would be you take the patient's skin cell and differentiate that into the relevant cell type for Parkinson's disease, for instance. And there's a company called Aspen Biosciences that's doing exactly that. They're trying to use the patient's own cells. So I think we're gonna see a lot more of that in the future as we start to learn what we should be doing therapeutically, then we can use people's own cells to get to that point. So,
Yeah, and related to that, as I mentioned that when you try to reprogram differentiated cells to iPS cells with these 4 factors, very few cells actually become fully reprogrammed to stem cell-like cells, right? But even the rest, there are also potential benefits. So the rest of the cell, they're partially reprogrammed, but not fully stem cell yet. So those cells typically, they look younger than the fully differentiated cells. So now there are actually companies and many labs that are using this technology to try to reverse aging, for example. So we have a new faculty who's gonna join UCI to work on that. So basically use different combinations of these factors, not to fully reprogram it to stem cells. We may not need that all the time, but to use that to partially reverse the aging process. And that's also a potential application area.
Dual mission, right?
Yeah, that's right.
I have a question about your CF 1125.
Yeah.
So if you block it, are there any like bad side effects, either in the cells or if it was blocked in a mouse or a mutation in a person?
Yes, so this is so you can imagine that when you block it, it becomes easier for cells to become stem cells. And the benefit is you make more stem cells, but the one pitfall is that stem cells in many ways are like cancer cells. So stem cells has 2 abilities, right? One is pluripotency, so they can become many other cell types. The other ability is self-renewal. So that means they can make more of themselves. So it's just like cancer cells, right? They can make more of cancer cells. And so that's why I think if you block CF125 for too long, then you might not only get stem cells, but you might even induce cancer. So that is a potential challenge. So that's why we wanted to develop this compound that you can just temporarily suppress it, but not suppress it for the long term. So let's say that, for example, mouse cells, it takes 2 weeks to reprogram. But the biggest effect for CF125 in this process can be observed in the first 5 days. After the first 5 days, even if you knock it down, it doesn't matter anymore. So then if we have this compound, what we can do is we can add the drug to differentiated cells for, let's say, 4 to 5 days. And then we take it away. That way you can get the benefit of improved reprogramming without the long-term harm, potential harm of inducing cancer-like cells, right? Yes. Yeah.
How did gene show tissue-specific gene expression pattern?
Yes, so during differentiation, so basically you compare stem cells and differentiated cells, they are different.
So my question actually is related to the differentiated cells.
Yeah.
Because if you have a cell line which already has a lower expression, I expect, based on what you said, the efficiency would be higher. Have you seen this kind of correlation, or
Yeah, so that we have, so for this one, for most reprogramming, we take fibroblast cells, right? So, and we have not checked whether different cell types can you get different level of increase by using different cell types. For example, in neurons, this factor is, the level is very high. And so potentially if you knock it down, you may be able to see bigger effect in using neuron to reprogram into stem cells versus other type of maybe highly proliferating cells in which these factor level is lower. And maybe when you knock down that in those cells, you will see less of effect. It's possible, but we haven't tested it.
But even in biologically speaking.
Yeah.
So we're just checking the expression of this factor
Uh-huh.
in the cells, which
Yeah.
has a lower one.
Yeah.
I would expect a high efficiency in terms of reprogramming. Or-
Yeah, I think it's likely, and it's one correlation is that usually the cells that are growing very fast, usually it's like stem cells, have lower levels of this factor. So that and so that this already shows that it definitely regulates growth, and so because of that, because of the starting levels are different in different tissues, then the benefit of knocking down for reprogramming can be different in different tissues. We just haven't tested extensively.
Do you know how many target genes for this gene?
Yeah, so that's basically hundreds. Yeah, and in some of them, we have shown that they function in differentiation or in reprogramming. And there's not a single like we didn't find a single gene, for example, that causes the most effect. But yeah, so that when you have that many targets, it's kind of hard to find always find the smoking gun.
Yeah, I can chat more. I know you have lots of questions.
So,
Yeah.
sorry, about a month or so ago, there was another lecture here at, ICI about organoids, I believe. That was also made using stem cells. And so with the current technology, do you think that it's possible to make a synthetic human or maybe even partially? And I was thinking that maybe like prosthetics.
Yes, so that's a very provocative question. So organoids, it's obviously right now that nobody's, well, as far as I know, nobody's trying to use organoids to make a whole human. But you can actually, there's a lab in Israel that's growing a whole animal outside the body. So basically, allow it to develop, right? So from an embryo into a whole animal outside of the body, so in a chamber. And so basically, yeah, you can potentially grow, for example, organs, entire organs that way, and use it for therapy. But I think I don't know if it's
10-year plan, right?
yeah. But of course, there are not only technology is involved, but also ethics is involved, whether it's ethical to do something like that, right? So there's all kinds of factors to consider, but I think that to grow organs is definitely possible. I think there's, for example, you guys probably have seen that people have grown entire ear on a mouse back, right? So, and you can grow these muscle cells in petri dish, and so that basically you can get organs and tissues from iPS cells. Yeah.
So, at like in a natural zygote, when it's in a totally potent state, how do the individual cells know how to differentiate into different cells? Like, is there any crosstalk between them? And is it based on the same mechanisms with the same factors and APA?
Yeah, so that's a great question. So this has been studied for a long time. That's a classic question in developmental biology. And so people have studied this best in using frog embryos, right? What people have shown is that even when the embryo, when the fertilization happens, so that's already depends on where the sperm enters the egg, and then it forms the fertilized the fused nucleus. And then there's the mothers and deposit different materials on different sides of the egg. that determines where, which side becomes the head, which side becomes the tail, which side becomes the back. And so there's spatial information already deposited in the egg cells. And then, so basically, once you kickstart the whole development process, that information is converted into developmental information. So basically, people have used frogs and used flies to study this. It's a very fascinating case. So it's obviously it's a combination of maternal deposited materials and the genes in the DNA. And so it's an interplay between the two. So there are mutations, people have used, for example, Drosophila fly to study this. And so they make mutations that can change development. So some of the crazy mutations, if you mutate one gene, the fly will grow 2 heads, right? So flies will grow 6 wings or something like that. So basically there are genes controlling all of this, but the initial spatial information that you're talking about is provided by the mother. They deposit proteins, RNAs at different sides of the embryo, and so that the initial spatial cue is already there.
Okay, well, if you have more questions, I'm sure Dr. Shi can answer some more with you and chat a bit more, but let's thank our speaker. That was fantastic.